All right. Good morning, everyone. My name is Dylan, and this is my colleague Cole, and it is a privilege to speak on behalf of the wonderful team at Queen's University, the rest of whom are sitting in the front row over there, for Mission 18 to the International Space Station. To begin, I think we can all agree that space exploration is perhaps one of the most marvelous but dangerous feats of human innovation. And in addition to all the technical and physical dangers that astronauts face on a day-to-day basis, a new threat is emerging. One where our challenges with antibiotic-resistant bacteria on Earth transition into space. For example, the bacteria E. coli has been consistently cultured from the air and from the surfaces of spacecraft. And the risk of severe E. coli infections only increases with evidence on how these bugs can be upwards of 10-fold more resistant to antibiotics in space. And if we compound this with the inevitable immune dysfunction that all astronauts will face, it is clear that E. coli presents a significant challenge to the future of space exploration. A challenge that warrants the need for more research to better understand how these bugs behave in space to inform the development of downstream solutions. And one aspect that makes E. coli infections really challenging to treat is the production of biofilms. And these are simply large adhesive masses secreted by bacteria that contain matrices of various cross-linked sugars. And given that biofilms already impede antibiotic entry as a physical and chemical barrier, their observed hyperproduction in space, combined with their ability to physically destroy hardware on spacecraft, illustrates a dire need for new solutions. Hence, our team was interested in looking at an alternative method to reducing E. coli biofilm formation on spacecraft. And one field that has continued to receive interest across all disciplines are plant-based solutions. And in our case, plant-based lectins have been consistently shown to reduce biofilm formation across a variety of different bacterial species. And specifically to E. coli, the plant-based lectin Con-A, which is isolated and purified from the jack bean, has been shown to inhibit biofilm formation by impeding the formation of those critical sugar crosslinks, either between sugar molecules themselves or between sugar molecules and bacterial cells. Hence, our hypothesis is that growth with Con-A will impact E. coli biofilm formation and composition under microgravity. And to address this hypothesis, we have three central research questions. The first simply seeks to understand how biofilm formation is impacted by Con-A in space. We then want to look at how the composition and structure of these biofilms may be impacted by Con-A in space. And lastly, we want to take a really broad look at how E. coli gene expression as a whole may be impacted by Con-A in space as well. And now hand off to Cole.
Thank you, Dylan. Upon arrival to the ISS, the lyophilate will be mixed with the preloaded medium of LB containing Con-A by releasing clamp A. And having the media pre-mixed with Con-A is an accepted standard for biofilm formation experiments in microgravity, as it reduces the technical complexity of the experiment without impacting the results. After 2 days, the cells will be exposed to RNA later by releasing clamp B. And RNA later was chosen as the fixative for this experiment, as it halts bacterial gene expression and growth and metabolism, preserving the cells for future genetic analysis. Once the sample returns to Earth, total cell count will be compared to our ground control, while gene expression will be analyzed using qPCR and RNA sequencing. In addition, the cells will be washed and resuspended in fresh media for future biofilm production and structural analysis. We anticipate that biofilm formation will decrease in microgravity due to Con-A-mediated interference with extracellular sugar chain crosslinks. This has been demonstrated in studies on Earth and in our preliminary ground truth experiments, which show Con-A inhibits biofilm formation at concentrations of 100 micrograms per milliliter and greater. However, genes encoding for cell adhesion receptors, such as CSGA and LRHA, may increase as a compensatory response to Con-A. Ultimately, using lectins to target biofilm formation increases bacterial susceptibility to current antibiotic treatments, which is a more sustainable solution than continuing to spend billions of dollars on new ones which will eventually become obsolete. We would love to thank all the national sponsors of the SSEP who help make this program available to students around the world, like us in Canada, as well as our amazing team of professors and lab research assistants who are always there to support us. Thank you.
Do we have any questions for this team? Okay, thank you very much.